All right, so today we're going to talk about productivity, ecosystem production, and this is in chapter 20. And I'm just going to start off by kind of reminding you what is required to be a living organism, what's required for life. The first thing is you need some kind of carbon source. All life on Earth is based on carbon, so that's what organisms build their biomass out of. Organisms then need some kind of electron donor. What do they use as an energy source? How do they synthesize ATP? And then you need an electron acceptor, which is basically what do you breathe? So it might seem kind of weird to talk about these life requirements in these terms, but there's so many different ways to make a living as an organism on Earth that it can kind of help to think about it in these three ways. And then, of course, you need water and you need space and you need to be able to reproduce and things like that as well. So for humans, our carbon source is organic carbon, the food we eat. Our electron donor is also our organic carbon food, and we use oxygen as an electron acceptor. So we breathe in oxygen, we breathe out CO2, and that's when we respire. So just kind of briefly, you can look at these slides with a little bit more detail. If you want to pause the video, you can see lots of different types of organisms here, and they're organized by their carbon source, their electron donor, and their electron acceptor, and then their breath byproduct. So what do they breathe out after respiration? And then they're classified by aerobic or anaerobic. So you can see humans, you can see what a photoautotroph does, you can see what organisms that are called nitrifiers or denitrifiers, or methanotrophs, which are organisms that basically eat methane, versus methanogens, who breathe out methane. Acetogens breathe out different kinds of organic molecules. And so here's some of the organisms. And then there's another slide that goes into more detail with even more diversity of chemoautotrophs. So sulfur oxidizers, iron oxidizers, sulfur reducers, sulfate reducers, manganese oxidizers, hydrogen oxidizers, and fermenters. So some of our most favorite organisms are fermenters, things like the yeasts that make bread, rye or beer. And you can see that fermenters use organic carbon as their carbon source, organic carbon as their electron donor. They actually also use organic carbon to breathe, and then their breath byproduct is CO2. So they make a lot of carbon dioxide, which is what makes beer fizzy. All right, so if you haven't spent a lot of time thinking about those things, you might spend a little bit more time on these two slides. So here's a picture from Yellowstone showing lots of different types of chemoautotrophs. You can see iron oxidizers around the edge where it's kind of rusty. You can see kind of a gradient of color. You can see lots of different types of organisms here based on the byproducts that they're producing through their chemoautotrophy. All right, so the chapter really gets into kind of classifying ecosystems by their energy flow. And this concept was really kind of poo-pooed early on. There was a paper in 1942 by a student named Ray Lindemann who tried to understand a lake ecosystem just focusing on energy transfer. And they really struggled to publish this paper. It was just too kind of outside the realm of what people were thinking at the time. But it has become kind of a foundational paper in ecosystem ecology, and pretty interesting way to think about systems. So we're going to talk about primary production mainly in this chapter, and that is chemical energy that's produced by autotrophs. And so you can either produce primary material through photosynthesis or through chemosynthesis, and that's kind of why I went into all those chemoautotrophs, which aren't really covered in the chapter. Primary productivity is the ultimate source of energy for all life on Earth. So although plants kind of are the ones that are called primary producers, they're producing this material. Their material and energy feeds all of their life on Earth, from the top carnivores to the detritivores. So it's a really important metric when it comes to understanding ecosystems. And ultimately, fossil fuels are derived from primary productivity from early on life. And primary productivity, I'm kind of using those terms similarly, but really primary productivity is the rate of primary production. So we talk about gross versus net primary production, and the reason is that gross primary production, or GPP, is the total amount of carbon fixed by autotrophs, and it's controlled by things like climate, photosynthetic rate, and leaf area index. Leaf area index is the amount of leaf area per unit of ground area. So that can be very, very small in places like the tundra, like 0.1, or it can be more than one. It can be up to 12, meaning that there are 12 layers of leaves for each, inch of ground area in the tropics. So here's something that a lot of people don't understand. So plants, of course, we know produce oxygen, but plants also respire just like us. So they breathe in oxygen and they breathe out CO2. That's something that a lot of people don't understand. And so about 50% of the carbon that's fixed by plants actually goes to respiration. It becomes CO2 again. And so that's really important. And that's the difference between gross and net primary productivity. So net primary productivity equals gross primary productivity minus respiration. It's basically the leftover carbon after accounting for respiration that can be used to build biomass, plant biomass, which is what those other trophic levels eat. So it's important. So NPP is related to carbon allocation. So once a plant takes up carbon through photosynthesis, it can be allocated to a variety of places. It can be used for growth or reproduction. It can be shunted above ground to produce above ground biomass. Say you're growing lettuce, that's where you want the carbon to go, or it can go below ground and make below ground biomass, things like roots. So if you're growing carrots, you want your plants to allocate a lot of carbon below ground to their roots. Plants, though, also allocate a lot of carbon to their symbionts, so especially things like mycorrhizae. So they'll leak a lot of carbon below ground to feed the mycorrhizae. And you can also, as a plant, you can allocate carbon to storage products, things like starches and tubers, or to defensive compounds that protect you from herbivores. So here's a picture showing the percentage of NPP that's allocated to roots, and you can see that only about 20% of NPP is allocated to roots in the tropics, but upwards of 70% NPP is allocated to roots in temperate grasslands. When you look at a temperate grassland, you're actually only seeing a fraction of the biomass, the majority of the biomass in a grassland, or, well, I guess that's not necessarily true, but a lot of biomass is below ground. All right, so if you can think of NPP then as the currency of an ecosystem, and so NPP is probably the most important measure of productivity, and because it's what carries up to the higher trophic levels, and it can be an indicator of ecosystem health, depending on what ecosystem you're looking at. And it's the carbon that's involved in the global carbon cycle. I guess all of it is, the respiration is as well. The ways that you can measure NPP are varied. You can measure the change in plant biomass in a plot, and you can scale it up to a larger area. You can measure fine root turnover below ground using rhizotron, like shown in this picture here. Or you can measure primary productivity for algae by measuring the chlorophyll concentrations in a slurry of algae. You can also measure productivity from space using normalized difference vegetation index, or NDVI. So vegetation has a really high NDVI value. This is basically looking at light reflectivity from the surface of the earth. Soil and water have very low values. And so we can use this difference between vegetation and soil or water to basically measure primary productivity from space. We can also use NDVI to measure things like desertification, pollution, CO2 uptake, and deforestation. So here's a picture showing NPP global values. So the kind of the more purpley bluey colors are really high values for NPP globally, and these data come from satellite imagery. Here's a picture of Mount St. Helens. We've just started using the same technology to measure productivity change through time since the eruption. And here you can see from 1984 through 2019, you can see an increase in NDVI, which is basically a measure of biomass. And I apologize, I'm noticing now there's no y-axis label on this graph, but take it from me, it's a measure of NDVI since the eruption. You can see a lot of variation for different locations across the pumice plain, kind of the patchiness of plant growth in this post-eruption landscape. And it was really cool because we could really quickly access these satellite data using Google Earth Engine and a collaborator named Christine Albano from Desert Research Institute. So NPP and productivity is also influenced by nutrients. And here you can see that it's influenced by nitrogen, phosphorus, and most importantly by the combination of nitrogen and phosphorus. So for both dry meadows and wet meadows, we see the biggest increase in above ground biomass when both nitrogen and phosphorus are applied to the plots. You can also see this in lakes. These are some pictures from some pretty old whole lake manipulations where they added nitrogen, they added phosphorus, and what we really found in lakes is that the phosphorus was the main limiting nutrient. Secondary productivity is a measure of the energy that's derived from consuming organic matter. So it's the productivity of the secondary consumers, or the productivity of the heterotrophs in the system, all the non-autotrophs basically. So the herbivores, the carnivores, the detritivores, and the omnivores. You can use stable isotopes to figure out what an organism is eating. So here we have a graph showing the difference between Pleistocene herbivores have more of a plant-based diet versus cave bears, which have more of a meat-based diet. You can see this jump of about 3 what's called per mil in the delta 15 in value, which shows you the difference between one trophic level and another trophic level. So stable isotopes can be used really nicely in some of these ecosystem ecology systems. Help to describe diet. And I know, So another kind of connection to some work that we're doing is we're looking at net secondary productivity, or you can look at net secondary production, which is a measure of the ingestion rate minus the respiration rate and minus the egestion rate, which is kind of waste products. And this gives you a measure of how much, like mass an organism can put on over time. And it really depends on food quality and animal physiology. These are some pictures of some stoneflies. We're using stoneflies as a model system at Mount St. Helens to try to measure secondary production. All right, so take a 10-minute break and come back for the next video.